And thank you all for being here. So you can understand, having heard Marshall, Summer, and Deb, that I would say at the outset it's been a real privilege to work with them. It's been working with Marshall for longer than I can recall. We started very young, so together. And you can also understand why I would say they're tough acts to follow, but I'm going to do my best. And I will provide, both of these fine physicians referred to the importance of families to them, sitting on the bench with them. And I'm here to represent the family perspective. Yeah. Okay. Next slide, please. So I'm going to break my introductory comments into two parts. My life before these three little boys and my life after these three little boys, or since these three little boys. Before these three little boys, I graduated from West Point, U.S. Military Academy in 1966, served on the Korean border between North and South, and then in Vietnam for a year, followed by going to Georgetown in Russian area studies, getting my master's degree in Russian area studies, and taught high school for 5 years here in Fairfax County. Then I went to the CIA for 7 years and got to study the East-West balance between the Soviet Union and the United States, between the Warsaw Pact and NATO. And then got to serve a wonderful 4 years at the U.S. Department of State, where I got to serve a lifelong dream come true. Got to serve on the U.S. delegation to Geneva, Switzerland, to help negotiate the INF Treaty, the Intermediate Range Nuclear Forces Treaty, which ended 2 whole classes of nuclear-armed missiles. Ours aimed at the Soviet Union, the Soviets aimed at our eastern and western allies, and we cut up all those missiles and launchers. That was a dream come true. Then I went to Capitol Hill for the first time. I worked with the House Armed Services Committee staff for 6 years. And across the hall was Congressman Billy Tozan's office, where my wife-to-be worked. So I met Rachel, my wife, in the hallway between Billy Tozan's office and my own. She had 3 little boys. They were 2, 4, and 6 when I met them. Perfectly healthy, we thought. But then and they had actually walked their mother down the aisle in our wedding in Shreveport, Louisiana, and all perfectly, we thought, healthy at the time. Just 3 years later, our middle son, Keith, then 11, was diagnosed at Children's Hospital, Children's National here in Washington, with Friedreich's ataxia. And we could spell it, but we didn't know a darn thing about it. So we went online, and we saw some really distressing characteristics or factors in this disease. We learned that it was life-shortening, degenerative, neuromuscular. It led to a reduced vision, hearing, and speech, curvature of the spine called scoliosis, requiring implantation of metal rods, first to get the spine straight and then to keep the spine straight. Also involved loss of strength and coordination in all 4 limbs. Your arms and legs began to function poorly, which led to using first a walker or a cane or the walls and chairs in our house, and then to a wheelchair. Our son went through all of that. He was diagnosed at 11. He got a scoliosis surgery at 16, went into a wheelchair shortly thereafter. And it also involves diabetes. About a third of our patients get frank diabetes. Almost all of them have some diabetic insult. also most condemningly leads to heart failure and therefore death in early adulthood. Our average life expectancy is early adulthood. We lost our son at 24, diagnosed at 11. He's this middle one, Keith. And he died with congestive heart failure. He had all the other symptoms, but he died with congestive heart failure. We also learned that night on our old Gateway computer, that sort of dates it, doesn't it? That there was no treatment. At the time, there were no clinical trials, very little research going on. And we had no medical experience to understand what was going on. There was also no organization devoted entirely to supporting research into this disease. There were a couple, like the National Ataxia Foundation, the Muscular Dystrophy Association, that had our disease as one of 42 or one of 35, and devoting very few resources to our disease and its research. But the good news was the gene had been identified almost a year earlier to the date. So that gave us some courage. So we agreed in 1998 to establish the Friedreich Ataxia Research Alliance. We collected a total of 7 parents, including ourselves. So 5 parents like ourselves, and 1 adult patient, and 3 scientists who had been devoting some of their time to research on Friedreich's ataxia. We were able to raise in our first year $100,000 by letters to friends and families, and 1 walkathon, and so forth. And almost half of that, a little over $40,000, came from the NIH, where we applied for what was called an R13 workshop grant to assemble the field. We knew from our 3 scientists what we should begin with was get everybody together. It had never been done before. There had never been a collection of the handful of scientists worldwide that knew something about Friedreich's ataxia. So we started with that. We assembled 65 scientists from around the world to join their 15 colleagues at the NIH. The common ground was all but 1 could spell Friedreich's ataxia, and he was the only one that didn't get a grant from us eventually. So that's how we got started, and on that 100,000, we kind of doubled that in the second year, doubled that in the third year, and so forth. And now we're raising a little over $12 million a year. 95% of that goes to research. We have very small overhead. We started all volunteers. My wife and I did the newsletters from the kitchen floor, so I was doing the science and she was doing the fundraising. But now we've just last November, conducted the world's 8th International Scientific Conference on Friedreich's ataxia, and that involved about 450 scientists instead of 0 drug companies at that first one. They all said, you're too rare, you're on your own. We now have more than 4 dozen scientists, 4 dozen industry partners. They're not only coming to our international conferences, they're helping us fund those international conferences, and instead of 0 clinical trials, by the time we did our 8th international conference, we'd already conducted more than a dozen clinical trials and completed them. We had another dozen underway, some of those clinical trials included what would be profoundly beneficial to us, protein replacement program that's in clinical trial now, 2 gene therapy programs, both for heart, and about a dozen gene therapy companies interested in conducting clinical trials in our disease. And on Rare Disease Day, February 28th, we got our first FDA approval. And a big step. We've got a long way to go. Trust me, this won't be profoundly beneficial. It will help our kids stay on their feet a couple more years while the gene therapies and the protein replacement strategies and the other small molecules come in for a combined therapy. So it was a grand first step. So how did we do that? How did we get to that point? Well, we've always taken, how many of you saw the Field of Dreams movie? You might remember, I think it was Kevin Costner who said, build it and they will come. We started with 0 drug companies. We could find 65 scientists around the world, all but one who could spell our disease. And now we've got, many, many more. And so we've built all that on the foundation. We built that Field of Dreams on the foundation of patients and their families. We built trust. We built hope, and then confidence. These people know we're going to get this darn disease, and we're going to get it together. We built it too on these dedicated scientists like Marshall and Deb around the world. We're giving them grants, and they're getting grants from the NIH to conduct the basic science, to understand and characterize our disease. It's so important. We've funded most of that, the basic characterization of the disease. They're also looking at mechanisms of damage, what's going on in the body, what's causing the damage, what the mechanism of action might be of a prospective therapeutic. So those are our basic scientists. We're also providing the translational tools, the assays, the cell and animal models, and the biorepositories. We've got blood samples, urine samples, cheek swabs, the whole deal. We also built the contact registry. We needed to know who our patients were, where they lived, how to get in touch with them. So we started with an Excel spreadsheet, and we built it up now to a global registry. We know who our patients are for the most part, the ones who have been identified. We know where they live, what their email addresses are, and we can use that registry to recruit clinical trials. We've used that patient registry, for example, to recruit a clinical trial of over 100 patients in 3 centers across the United States. We did it in 2 hours and 48 minutes. It sounds ridiculous, but because so many trials fail because of lack of patience. But that's our clinical, our contact registry. We've also developed consortia for biomarkers, the biochemical ingredients in our bodies that signal success or failure, signal you're getting worse or you're getting better. And a very important project in neuroimaging. These are scientists around the world who are using MRIs and MRSs, these magnetic resonance imaging. You've seen the videos probably of people being slid into this massive machine, and they're using these images to determine very important things like what is the damage being done in this disease, where is that damage being done, and when is that damage being done, so that we can target these prospective therapies at not only the right places, the right tissues in our bodies, but at the right time. So very important project that we're funding with the help of these industry partners. It's a collaborative, pre-competitive project. And very importantly, I'll finish that section with this, our natural history study. How many of you have ever heard of a natural history study? Yeah, Marshall knows a lot about it. Stuart knows a lot about it. These are studies of patients without intervention. You get the patient into your clinic, you examine that patient to try to determine what's going on. What's the symptomology? When is it developing? You're using examinations to determine what's going wrong and when is it going wrong and at what rate does it progress. And you can use that to determine, our drug companies are using it now to determine what's the characteristic of the patient that's optimum for my therapeutic approach. and what age is optimum for my therapeutic approach, and what are the procedures that will be best suited to these patients. How long can I examine them before they get so tired or they need to go to sleep, and so forth. And so they can combine that natural history study that we've been conducting for over 20 years. So we've got like 3,000 patients who have been through our natural history study. They go to their clinician once a year to determine the rate of progress. And until February 28th, we had no therapy to offer them. So it was all natural history. There was no intervention. So that's been a very important aspect for us. And now all those data from all those exams, from those thousands of patients around the country and around the world, are on a FDA-funded program called the Rare Disease Cures Accelerator Data and Analytics Platform, RDCA DAP. And that's a program that's implemented by the Critical Path Institute with help from the National Organization for Rare Disorders, and again, funded by the FDA. We are a leading disease in that platform, and we are strong proponents, as is Marshall, of that platform. I will tell you without reservation or hesitation that that platform in our natural history database is the reason we got our approval on February 28th. Because that database is readily available to the FDA. We've had several meetings with the FDA with our natural history data in front of them. They not only knew that natural history, they trusted that natural history. It's high value. So they knew when we showed them that, and this is, I'll skip ahead here. I'd been working and knowing one of your colleagues in journalism, Amy Marcus of the Wall Street Journal, Pulitzer Prize winning journalist that won the prize based on her work in Niemann-Pick C, another rare disease. I've been talking with her for several years. She always told me, very importantly in journalism, especially the Wall Street Journal, we will not, we love what you're doing, but we will never write a story about a success in a single rare disease, no matter how important it is to you. What we want to see is that your success has broadly based impact on all rare diseases or on many rare diseases. And guess what? She saw what we had done with our natural history study and convincing the FDA, who until 1999 insisted on more than one clinical trial in a rare disease to give you approval. They always said it was adequate, well-controlled studies is what the regulation said. You had to have more than one. Beginning, I think in 2017, they started loosening that a little bit and said, okay, if you can do one adequate, well-controlled study, complemented by confirmatory evidence that the study results are valid, then we will consider approval. And at the same time, the guidance said, here are the ways you can provide confirmatory evidence. One of them was natural history, but they had never found that sweet spot of sufficient natural history to give a disease group approval based using natural history. They continue to push back on using the natural history within the clinical trial, to supplement the cohort that's called the placebo. So some of these patients are on drugs, some of these patients are on a placebo, and they have really resisted using natural history to substitute for that placebo. Say, here are a bunch of patients that aren't taking the drug. Why can't we use them? Well, it's too complicated. But in this case, they said in their guidance, you could use natural history to provide confirmatory evidence, but they hadn't done it yet. So we come along with this really high-value natural history database, and we provide confirmatory evidence that in the continuing open-label extension, clinical trial is completed, results are collected and submitted, but the open-label extension, everybody's on drug, no more placebo. During that 3 years that these patients were on open-label extension on that drug, we showed, comparing them, the 137 patients in that open-label extension to 137 very carefully matched patients in our natural history database, we showed a 55% improvement compared to that natural history. They said, that's compelling. That's why they gave us our approval. So, and that's why Amy Marcus published an article in the Wall Street Journal the night that we got our approval. So February 28th of this year. So thank you, Amy. Next slide, please. So I've talked about, well, first back to relationships. If you could go back a slide, please. Common statement here in Washington, I'm sure it's the same in San Francisco, it's all about relationships. You know, you make one mistake in these relationships, you'll never get a job in this town again, sort of thing. And as I said, we've built our whole foundation on the trust and of our families and the confidence of our families and the scientists and the NIH and the FDA. Because Joni wasn't able to make it due to the chicken pox, I'll emphasize the NIH because I've had the good fortune of serving at the NIH on the what's called the National Advisory Council. Every institute and center has a National Advisory Council. Marshall now serves on the NCATS Advisory Council with, that's Joni's center, the National Center for Advancing Translational Sciences. I got to serve on that advisory council. And I also got to serve for 4 years on the Neurological Institute's advisory council. Wonderful opportunities to see what the NIH is doing for all of us and what NCATS in particular has done for ours. We've got 3 drugs in clinical trial that have benefited from NCATS, and one of them is a gene therapy trial. So we're really profoundly thankful for help from the National Institutes of Health. I've also been able to serve as recently, I'm the co-founder of the NCATS Alliance, the National Center for Advancing Translational Science Alliance. Put in a little bit of a plug for that because we've just sent our first request to Congress to increase the budget at NCATS, primarily because, you know, we all know about gene therapy. We all know about what a promising approach gene therapy is. We run the risk of losing that opportunity in gene therapy because now company after company is abandoning gene therapy in favor of returning to small molecules because it's proven to be much harder than we all anticipated. We got the first news about gene therapy program. We said, we just cured the mouse, the FA mouse with gene therapy. We said, well, this is going to work. We heard from our basic scientists, this is really going to work. It's proving to be much harder than we hoped. And now companies, because I think Marshall and Deb emphasized the expense. Hundreds of millions of dollars to develop a gene therapy program, and great risk. There are gene therapy programs. There are only about 25 gene therapies that are approved. More than half of them are in cancer and blood disease. There's only one central nervous system approval with spinal muscular atrophy. So it's much harder than we hoped. And we're losing that opportunity. That's why the NCATS Alliance is so intent upon getting the NCATS, getting the NIH to solve some of the 5 basic problems that are confounding progress in all these gene therapies. So next slide, please. I'll just summarize this quickly. I know I've taken too much of your time. So we've done enough for all these companies, giving them what I call what they need to succeed. You know, all the background information, all the natural history, the translational tools. And so they come to us early and often, before they even have a drug, before they even know what tissue they want to amend. And they know they can work with us and our key opinion leaders and our natural history study and our patient registry. We will give them their best possible shot on goal. You know, their drug may fail the trial, but the trial won't fail the drug. And working with us will not fail the drug. So next slide, please. So the conclusion is that all these sponsors tell us they've chosen our disease because of all the things that we're able to do for them and our willingness to do it for them. They've become convinced that, as I said, the clinical trials they design with our help and the help of our natural history will not fail their drug. And they know that we will give them the best shot on goal and that some of those shots will find the back of the net. That's what we're about. So we've got a long way to go. We're convinced with this first approval that we've got the pathway to get there together. So thank you very much for your time.